Today's topic is about Spina Bifida. But first, let us start with a question. What is Spina Bifida?
Spina bifida is a congenital malformation in which the spinal column is split, often with spinal cord involvement, as a result of failed closure of the embryonic neural tube during the 4th week post-fertilization. There are 4 types of spina bifida. Occulta, which is the mildest and most common form, in which one or more vertebrae are malformed and covered by a layer of skin. Closed neural tube defects, where the form consists of a diverse group of defects in which the spinal cord is marked by malformations of fat, bone, or meninges. Meningocele, in which the spinal fluid and meninges protrude through an abnormal vertebral opening. The malformation contains no neural elements and may or may not be covered by a layer of skin. Myelomeningocele, which is the most severe form, occurs when the spinal cord are exposed through an opening in the spine, resulting in partial or complete paralysis of the parts of the body below the spinal opening. In this video, we will be focusing on the myelomeningocele. The caudal end of the spinal neural tube fails to close during embryonic development, which results in the spinal cord being open dorsally. During prenatal development, neuroectoderm thickens into the neural plate, which then folds into the neural groove by the time somites appear. The groove deepens to become the neural tube, and dorsal fusion begins centrally, extending cephalid and caudally, with the cephalid pole fusing at the 25th day. The ventricle becomes permeable at the 6th to 8th week of gestations, but this apparently does not proceed normally in patients with MMC. The opening forms a placoat on the back of the fetus or newborn baby, which frequently rests on a meningeal sac or spina bifida cystica, containing dysplastic spinal cord, nerve roots, meninges, vertebral bodies, and skin, and the vertebrae at the end of the lesion lack neural arches, resulting in it being dorsally incomplete due to the exposed neural tissue being degenerated in utero. Neurological, motor, and sensory deficits that varies with the level of lesion are shown. 90% of infants with MMC suffer from deformation caused by the development of the cranial neural tube, called Cary type 2 malformation. Normal intelligence can be expected with aggressive shunting for hydrocephalus. Other effects of MMC include neurologic damage, which will result in neurogenic bowel and bladder, leading to incontinence, multiple system congenital anomalies such as facial clefts, heart malformations, and genitourinary tract anomalies, obesity, bone involvement, urinary tract dysfunction, and latex sensitization. MMC is one of the most common congenital malformations. The cause of it is still largely unknown. Some studies suggest that an increased amount of neural crest material in the defect prevents neural tube closure. Another hypothesis is that an already closed tube ruptures. Increased permeability of the rhombic groove leads to the greater CSF secretion and increased luminal pressure, with the tube then expanding and essentially splitting the neural element at its weakest areas.
Internationally, the highest rates of spina bifida are found in parts of the British Isles, mainly Ireland and Wales, where 3 to 4 cases of myelomeningocele per 1,000 population have been reported. The reported overall incidence of myelomeningocele in British Isles is 2 to 3.5 cases per 1,000 births. Low socioeconomic status is associated with higher risk of neural tube defect in many populations. Since approximately 1940, epidemics of myelomeningocele have occurred in Boston, Mass., Rochester, New York, Dublin, Ireland, the People's Republic of China, and Jamaica. According to the CDC, the prevalence of spina bifida in the United States is higher in the white and Hispanic populations than in the black populations. around 1983 to 1990, the birth prevalence rate of myelomeningocele to be slightly higher in females than in males, which will exhibit thoracic level malformation. Nearly 14% of all spina bifida neonates do not survive past 5 years of age, with the mortality rising to 35% in those with symptoms of brainstem dysfunction secondary to the Arnold-Chiari malformation. While 70% of patients have an IQ above 80, only half are able to live independently as adults even with adapted accommodations. Spina bifida have appeared from around 1200 BC, and from skeleton that is unearthed in Morocco showed evidence of spina bifida. In fact, references to the conditions have been illustrated and documented throughout history. Some of the references paved the way for the 20th century pioneers of science and medicine. 2 mummified children were found in the tomb of King Tutankhamun. It is believed that his wife, Ankhesenamun, had 2 girls who both died at birth. X-rays revealed signs of spina bifida in both children. Hippocrates, the father of medicine, was the first physician to document and treat hydrocephalus. He believes he can cure what he called as water over the head with foot and small hole drilled into the front of the head that is called as trepanning. On 24 August 79 AD at 1 p.m., Mount Vesuvius erupts and kills everyone in Pompeii, burying them under tons of ash. The remains of 2 young children are found with evidence of spina bifida. Greek physician, surgeon, and philosopher Galen mentions hydrocephalus several times in his written works. On his proposal of treatment, he used tree bark and twisted it around the patient's hand and inserting it into the trepanning's opening. Not surprisingly, this doesn't work. Paul of Aegina is one of the last great Byzantine physicians that proposes that one of the possible causes of hydrocephalus in infants could be down to their heads being squeezed too much by midwives during birth. On the Dark and Middle Ages, any references to spina bifida or hydrocephalus come from Islamic medicine. Al-Zahrawi is the father of surgeon and currently considered to be the greatest medieval surgeon from the Islamic world. He writes his 30 volumes medical encyclopedia, which contains several mentions about diagnosing and treating hydrocephalus. He includes the first detailed description of how cerebrospinal fluid could be drained using one or multiple T-incisions in the head. Elizabethans are obsessed with the birth of misshapen human beings and animals. They shared this fascination through several sensationalist publications and journals. One such image, the form and shape of the monstrous child born at Maidstone in Kent, show a small child with spina bifida significantly exaggerated for the readers. Dutch surgeon Nicolaas Stolp is the first person to use the term spina bifida. He talks of an occasion where he treats the condition by puncturing the sap on the back. Unfortunately, the patients died, but he discovered the presence of a neural element entering the sac. This paves the way for a more sensitive surgical treatment. Giovanni Battista Morgagni is the first person to fully describe hydrocephalus and make a clear connection to spina bifida. He proposes that spina bifida could exist with or without hydrocephalus. Rudolf Ludwig Karl Virchow, known as Pope of Medicine amongst his colleagues. He is the first person to describe and start using the term spina bifida occulta. A German surgeon, C. Bayer, performs the first surgery repair on an open myelomeningocele, the most common form of spina bifida. He uses an innovative method of a series of rotating flap techniques with moderate success. It is considered a remarkable advance and is sure to be adopted by many other surgeons.
Let's talk about the trending research about spina bifida. The most trending research is about placenta mesenchymal stromal cells, a potential treatment for spina bifida. Placenta-derived mesenchymal cell is derived from early gestation placenta, possesses all of the phenotypic characteristics of mesenchymal cells, and secrete high levels of the neuroproductive, immunomodulatory, and angiogenic factors including brain-derived neurotrophic factors, hepatocyte growth factors, VEGF, and the chemokines monocyte chemoattraction protein 1, interleukin 8, interleukin-6, and the IMP1. PMCs have paracrine functions which their secretome has a profound effect on the recovery of apoptotic cells. Therefore, PMCs can increase the number of proliferations and recover the cells that are undergoing apoptosis from complete damage. Another current research of Spina bifida is about recent advances and future perspective. Direct interactions of the downstream target sites between the responsible regulating genes unique pattern of biology and genetic roadmap control the new relations and provide further insights for the causes of Spina bifida and play a role in the development of new molecular targeted therapy. For example, the importance of molecular cytogenetic technologies such as array comparative genomic hybridizations and fluorescence in situ hybridizations to clarify the unbalanced chromosomes rearrangement, which is one of the positive sides of Spina bifida investigation.
One of the treatment for myelomeningocele is called MYOM or management of myelomeningocele. So this MYOM is divided into two types, which is prenatal surgery and postnatal surgery. For the prenatal surgery, the surgery is performed without removing the fetus from the mother's womb. So the neural tube and the other layers of the back are surgically closed by neurosurgeon when the fetus is still in the mother's womb. After the procedure, it all closed and also the womb of the mother is closed. This prenatal surgery results in decrease of fluid inside the brain, improvement in motor neuron, increase the possibility of the child to walk unassisted, and restoration of some nerves. However, there is some disadvantage, such as increasing the risk of spontaneous rupture of the membranes, oligohydramnios, and preterm delivery. The outcome of this surgery is mainly for secondary problems, which is treat the Chiari malformation or the cerebellum malformation and hydrocephalus, but it can still cover the primary problems such as paralysis and bowel problems. Next, we will talk about the other part of MYOM, which is called postnatal surgery. So, postnatal surgery is a normal surgery which performed after the child is birthed. The purpose of this surgery is to cover the exposed spinal cord to prevent infection. In this surgery, the cerebrospinal fluid will be drained from the brain using ventriculoperitoneal shunt to the stomach. But however, the child will be paralyzed because the nerve is already formed inside the body. So, it will only recover the secondary problems, which is the cerebellum malformation.
Thank you for watching our video. Please give us like, comment, and don't forget to subscribe.